All right, so let's get started. I'm super happy to introduce Tom Miller. It's been great having him here. He has to go and teach, so he's flying out this evening. I'll probably try to, we have room for maybe one or two more if we, stop off somewhere on the way for a little after the seminar. So Tom, I knew about Tom's work and I knew he did cool stuff, and I sort of recently met him. We happened to be on the same NSF panel, and it was that funny kind of experience where it was all on Zoom, which was awkward. But I'm sitting there getting ready to read my thing, and like it's okay, you two rated this pretty high, and so who's going to go? And Tom started speaking up, and I was like, and he kept saying, and I was like, it's exactly what I was going to say, and I was like, I kind of realized that we had sort of very similar viewpoint, and so it's been really nice to in person having some great discussion. I think he has broad interests that overlap with a lot of the department, and as you know, a very strong group over in Rice. So it's really nice to have these interactions. Tom is PhD in Nebraska, Lincoln, is associate professor at Rice, and works on a bunch of different topics from theory to the field work and everything in between. And I'm excited to hear what he's going to talk about.
Thanks, Tim. And thanks to everybody for having me. It's probably been 10 years or so since I last gave a talk here. So it's great to be back. A lot's changed, but it's great to see some old friends, make new friends. So thanks so much for having me. Let's see. Okay, so I want to start just by orienting us in scales of organization. So thinking about individuals to ecosystems. I am a population biologist, so I think a lot about the spatial and temporal dynamics of populations and the ways in which individual level processes give rise to population level patterns. And I'm particularly interested in connecting population patterns to environmental variation, so biotic interactions and abiotic drivers that might vary in space and time. And that, of course, is like the founding objective of ecology. So it's not particularly novel in that sense, but it actually remains at the forefront of ecology because we live in a rapidly changing environment. And this is a data visualization that you might have seen, just representing the ways in which our climate has changed over the last 150 years or so, getting to our present very warm state. So this is not a climate change talk, but I wanted to start here to ground the idea of environmental variation. And if we can understand the environmental drivers of population level patterns, we might be able to predict consequences of environmental changes like we're seeing. So I'm going to use this idea of environmental variation to frame kind of two parts of this talk that will focus on the two dimensions of variation, which are space and time. So as Texans, we all probably have some visceral lived experience with environmental variation. So we can think about spatial variation. If you've ever driven from Beaumont to El Paso or vice versa, that's you've crossed one of the sharpest continental climate gradients on the planet. So very different conditions from one side of the state to the other. In time, we've also got a lot of variation. So this is a time series of 20 years of the area of the state of Texas that are in different stages of drought. So if you lived here in 2010 or 11, you know what that was. That was the very intense drought that we had. So we've had some good times and bad times, and really anywhere you live, you'll see dynamics like this. But for those of us who live here, this is what it looks like. So I'm going to use these spatial and temporal dimensions to frame two parts of this talk that will really tell you two stories. One about how spatial environmental variation generates range limits of species, and the other about temporal environmental variation. And I'll talk about plant microbe interactions in temporally varying environments. All this work will focus on plants and specifically grasses, which are a prominent study organism that we work with in my lab. Before I get into the details of this work, I want to start by talking a little more broadly about demography, which is foundational to a lot of the work that I'll present. So, as a population biologist, I think a lot about individual to population scale processes, and particularly the idea that populations can be structured by different demographic groups. So in this example, there's baby penguins and grown-up penguins and male penguins and female penguins, et cetera. So there's many ways you can slice up a population, and individuals in different demographic groups can exhibit very different vital rates. So rates of growth, survival, and reproduction. And that's an idea that has a long history in population biology, and we can draw out a life cycle diagram that represents how individuals might move through some cartoonized life cycle here according to these rates of growing, surviving, and reproducing. And we can convert a cartoon life cycle to a mathematical statement that says how a population structured by some state variables which appear in this vector, how those get updated through time according to a projection matrix that summarizes all of the paths through a life cycle. So in practice, what we do is we collect data that informs these individual level paths through life cycles. And from that, we can derive estimates of the population growth rate, lambda, or equivalently population mean fitness. And particularly in the context of environmental variation, we can connect vital rate variation to environmental data that allows us to understand how population viability might vary as a function of the environment. And we can identify thresholds in environmental conditions where populations may reach some viability limit. So in this case, when the growth rate falls below 1, there's more deaths than births and the population cannot persist. So this is a very quick and cartoonish overview of the nuts and bolts of how we do this work, but this will be kind of under the hood of a lot of the results that I'll show you. Okay, so I want to start by talking about this first element of spatial environmental variation and geographic range limits. This is work that was done in collaboration with my colleague and former postdoc Aldo Campagnoni. Aldo's now at the IDIV in Leipzig. And this is work focused on drivers of range limits. So this kind of gets at one of these core ecological questions of why are species distributed where they are and why are they not where they're not. And we have longstanding ecological theory that helps us understand that problem. Okay. So we can think about a species range as a manifestation of ecological niche, which we can represent as how population viability of a species might vary as a function of the environment. And canonically, we might expect some hump-shaped relationship that represents good environments in some intermediate range and then stressful environments as you move away from some optimum. And we can expect range limits potentially to arise at the limits of environmental variability, assuming dispersal is sufficient that everybody can get around to the places that they can live. We might expect in this example limits to population's viability at some upper and lower elevational limits. So there's this longstanding idea that niche limits can tell us about range limits. And of course, as species ranges are shifting in our changing environment, there's a greater urgency to understanding how range limits might arise. I'm going to suggest that this idea that range boundaries can be interpreted as niche boundaries and vice versa intersects a little awkwardly with another pervasive topic in ecology, and that is intraspecific niche variation. So this is the idea that if we go back to populations consisting of multiple demographic groups, not only do those groups differ in their demographic vital rates, but they may also differ in niche characteristics. So the ways in which they interact with the biotic or abiotic environment. So this is represented in a lot of work in ecology, including these a couple of influential papers. Why is this an awkward combination? Well, if there's many niches in a species or a population, then we've got something that looks like this. So then if we want to understand how niche limits translate to range limits, whose niche is it that governs the geographic distribution, and how would we know? So that's really the work that motivates this particular project. I'm going to talk about a specific type of intraspecific niche variation, which is sexual niche differentiation, which is where males and females of the same species exhibit ecological differences, so different resource requirements, different habitat use, different environmental tolerances. This is really well documented in all kinds of animals. So, I'm showing a couple of cherry-picked examples of birds and mammals and reptiles. And of course, it's animals that are sort of most associated with dioecy, or the separation of male and female function, but plants are dioecious or can be dioecious. It's taxonomically infrequent. So it's about 5% of angiosperm genera are estimated to include dioecious species. So they're not very common taxonomically, but these often are ecologically important species. So foundational species in some sense. So all of the willows and cottonwoods, junipers, hollies, many grasses are dioecious, having separate males and females. So I'm going to focus on dioecious grasses specifically. I'll circle back in a few minutes to these other woody dioecious groups. So if we've got sexual niche differentiation, then this is the picture that we're working with hypothetically of, just two niches, which is a little more tractable. So we've got males and females that potentially could respond to their environment in different ways. And if we're going back to the question of whose niche is it that determines the geographic distribution, we actually have an answer, or we can assume an answer, which is females. And that's because almost all of ecological theory assumes female dominance. So if you've ever written DNDT, the N is females, either explicitly or implicitly. And that makes a lot of sense. Females are their regenerative sex. And, so we're assuming that the availability of males has no influence on female fertility, which is of course wrong, but it might be often adequate. If there's not huge variation in sex ratio, maybe that's a good enough assumption. Our question is, if you have sexual niche differentiation, then you expect skewed sex ratios approaching range edges. And that might be a circumstance in which that's a bad assumption. Maybe you have mate limitation that arises as you approach range edges in ways that make it important to explicitly account for both sexes and their independent responses to those environments. So we can ask that question a little bit more formally if we think about, first of all, a female dominant perspective that might predict some environmental driver that affects female vital rates, growth, and survival. And in a female dominant perspective, we can derive some expectation for a niche response to environmental variation and how range limits might emerge from that. So that's one competing hypothesis. But the other puts males into the picture and says, well, males might have a different response to the same environment in ways that affect the composition of the mating pool. So if males and females are doing different things in response to a shared driver, that could affect the availability of males and females in the mating pool. And that could have a feedback to female vital rates and specifically fertility. That feedback can then generate this other pathway through which environmental drivers might affect population viability. So our goal was to quantify these sort of competing pathways through which the environment can affect population dynamics of dioecious species. So to do that, we worked with this model species. So this is Texas bluegrass. It's our namesake bluegrass. If you didn't know, we had a bluegrass. This is it. It's a dioecious species that occurs throughout the southern Great Plains. It's endemic to Texas, Oklahoma, and Kansas. This map is showing, first of all, the precipitation gradient that I talked about earlier going across longitudes. And the hatched shapes are the county occurrences of this species. So it sits right on this really sharp part of the precipitation gradient, suggesting that precipitation might be an important driver of its range limits. It's a species with genetically based sex determination, and it's a very tractable plant to work with. We can clone out males and females that we know the sex of and then generate lots of plants that we can work with experimentally. One of the first things we did was just survey the sex ratio across this environmental gradient. I'm going to focus a lot on longitudinal variation because that's where this strong environmental gradient sits. And we found, first of all, a lot of noise. There's, some male-only populations, some female-only populations. This is the operational sex ratio. So we often don't know how many genetically individual female and male plants there are, but we know the composition of the flowering population. So on average, we tend to get male biased flowering populations in the west and female biased in the east. Okay. So that was some early indication that there might be some kind of sex specific responses to this environmental driver. So our objectives then were to test those responses experimentally, to quantify the demographic effects of sex ratio. So the feedback here from the mating pool to female vital rates, we wanted to quantify that, and then put it all together to understand what are the drivers of range limits and what are the contributions of both males and females to that process. So I'm going to talk about the second thing first. I should have reordered this list, but I'm going to talk first about how to understand the demographic consequences of variation in sex ratio.
Excuse me, would you define mating pool?
Mating pool is basically relative abundance of males and females.
Okay, the second.
Sex ratio. Yeah. So I didn't want to use sex ratio because I'm really talking about operational
Yeah,
sex
yeah,
ratio.
got it.
Yeah. So we did this with a response surface experimental design, which is represented here. So these aren't results. This is actually an experimental design. So we can create experimental populations that vary in the relative abundance of males and females, operational sex ratio, mating pool, whatever you want to call it. So as an example, there are some treatments here that would look like this in the field. So this is mostly males, mostly females with one male. So the white tags here are females, and there's the male, the red tag. This is about a 1 meter by 1 meter plot. The opposite, looked something like this. So lots of males with few females. This was an experiment done at one site in the center of the range. And to get straight to what we learned here, this is what it looked like for seed viability of females in these different sex ratio environments. So starting with on the left side of this graph, where the operational sex ratio is very male dominated. So the rare female surrounded by males has maximum fertilization at about 85% of of the female's ovules are fertilized. If you go to the other side of that graph, where it's nearly all females, when there's no males, there's no seeds. And there's a ton of zeros that are sitting on top of each other there. So that's not particularly surprising. But we can sort of identify there's some limit at about 75% female where fertility really declines. So there is sex ratio sensitivity, but the females are pretty robust in terms of their seed production to availability of males until you get to about 75–80% female. So that's one piece of it. We now want to sort of go back to this first thing. So how are females and males responding in their demography to environmental variation across this longitudinal axis? To do that, we did a big experiment. And so every black dot here was an experimental site. And I talked a lot with some of you about field stations and the value of field stations, how we have all these wonderful resources, particularly in Texas. And, so this is Bashkot, Stengel, Lost Pines. So thanks for having us. This is the Texas Tech Field Station in Junction. We have wonderful resources really across Texas, none of which are Rice's, because Rice doesn't have any field station facilities. So I'm so grateful for our colleagues and friends across the state who welcomed us to do this work. So we worked kind of all over the place in Texas, Oklahoma, and Kansas. The other thing happening in this figure, there's a lot going on, but the red dots are the source populations that we drew from to create these experimental gardens. So you could imagine there's also a local adaptation type of question that you could ask here, which we're doing, but I'm not going to talk about. So we've got 16 sites across these 3 states, 12 plots per site, 6 plants per plot, 3 males, 3 females, and we did this for 3 years. And to get straight to the results, remember that we're looking for vital rate variation and how males and females might differ. And for most of what we measured, vital rates were pretty similar between males and females, showing a peak in the middle of the longitudinal range, which is sort of the classic expectation that the middle of the range is the best place to be, and not much difference actually between males and females. So not super strong niche differentiation, with one exception, and that was flowering. So this is flowering data for small, medium, and large plants. Don't worry too much about the size differences, but here what I'll emphasize is that there was a sex difference, particularly in the eastern part of the range. As you go east, females outflowered males. So there was still an intermediate longitude at which flowering peaks, but there is a female reproductive advantage, so the female reproductive niche extends further to the eastern parts of the range than the males. And we can actually, or we did recapitulate the pattern that we found in the natural population. So I showed you this earlier. This is what we see just in natural populations across that longitudinal gradient. In our experimental gardens, which is shown on the right, we got, or we started to see female bias flowering, female bias sex ratios in the eastern part of the range, 2 or 3 years into this experiment. Yeah.
It's an emergent pattern. You put plants there, like equal males and females, and
Yes,
Just
yeah, so you
sometimes.
plant equal numbers of males and females, and then, a year or two later, measure the sex ratio of the flowering population. So we get, as we saw in the natural population, more female flowering in the east. We didn't quite get the same male bias trend in the west, and there's probably lots of interesting reasons why that is that we're not totally sure about. But we do think we learned something about what generates this pattern. And it's not a survival or difference between males and females. It's just that females flower better under the mesic conditions in the east. So going back to kind of these ideas, what we learned so far was that probably both of these things were happening. So we do know that, for both males and females, the middle part of the range is the best. And as you go to the edges, it's just harder and harder to live there. So there's probably some element of female dominance where it's just, everybody suffers as you approach range edges. But it's also the case that sex ratios get increasingly skewed as you approach range edges, and particularly at that eastern edge, females are going to outflower males and have the potential to be mate limited if you get into very female biased populations out there. So we now wanted to know, okay, if both of these things are happening, can we quantify their relative importance? So to start, we can sort of estimate the population viability from all of our demographic data across our longitudinal gradient. And that's shown here represented with the uncertainty bands that come from our statistical analysis. And kind of as you might expect, there's this peak in the middle. So the middle of the longitudinal range is the best place to be. And what we were excited about from this is that if you look at the observed range limits, and so these are the westernmost and the easternmost places where we know these plants to occur, they're actually pretty well predicted by this model. So the uncertainty intervals are kind of crossing the threshold of population viability at around the place where the real populations kind of hit their limit. So this was not fit to that distributional pattern. It was independent data. So we were pretty confident that the model was telling us something real about the biology that generates these range limits. The last thing we can do is that decomposition of what are the females contributing versus the males. So this is a decomposition of the female contributions to this pattern. And the way to read this figure is that the thick black line is the total contribution of females through all female vital rates. And where this relationship is steepest, this line is highest or lowest. So this is the derivative of that. So this is telling us that this blue line is female growth. So female growth makes the greatest contribution among the female vital rates to why population viability declines, going too far west or too far east. So that's females. So now we want to compare that to the contribution of males. And this is the big reveal. And males do nothing, basically. So this is a real result. There's almost zero contribution of males to range limits. So the female dominant hypothesis that I started with, that wasn't a straw man. That actually describes the system really well. So why is that? Well, we think that because all vital rates tend to decline approaching range limits, females have other problems by the time that they get out to the eastern limits. So it's true that they're mate limited at their range edge, but that's not their biggest problem. The biggest problem is that they're not surviving well, they're not growing well. So they can, I don't say they can tolerate mate limitation, but it doesn't really register much in terms of the overall fitness decline. So we can take this big thing, which is nonlinear, and there's feedbacks between population structure and vital rates. And that's partly why people like female dominance, is because it's hard to work with two sex models. And our results show that you actually can simplify this to a really kind of simple linear system that doesn't worry about males at all. Where we're kind of, well, so to sum that up, so we do find evidence for sexual niche differentiation across geographic distributions. There are biased sex ratios at range edges, but it doesn't strongly contribute to the range limitation process. It's really a female dominant process, and female responses to environmental variation tell much of, if not all of, the story. Where we're trying to go next with this is kind of going back to some of those woody dioecious plants that I was talking about, which, as I mentioned, are often foundational habitat forming species. So we're interested in the unique responses of males and females to climate change. So thinking about climate variation in time and how climate change might perturb population sex ratios of these foundational plants. And we're trying to also connect that to the micro and macrobiota the microbes and the arthropods associated perhaps differentially with males and females. So to understand part of the biodiversity consequences of shifts under of sex ratio under climate change. And we're doing this as part of a new project through the Biodiversity on a Changing Planet program at NSF. And it's a binational project. So we've got collaborators in China, and we're developing Texas and Sichuan province comparative studies with these pairs of congeners where we've got species in both countries. And the goal of that is that, Sichuan province is, nearly the size of Texas, occurs at the same latitude as Texas, but they have very contrasting regimes of climate change. So we're trying to generalize our insight into, sex ratio perturbations under climate change by covering lots of species in very contrasting climate change environments. I don't have any data to show. This is really just starting up, but part of why I mention it is because we're looking for students and technicians to get involved in this work. So if you know of somebody who might be excited about this kind of stuff, please reach out to me or have them reach out to me. Okay, I want to shift gears to talking about temporal environmental variation. So all of that, or just about all of that, was thinking about how the environment can vary through space. And of course, the environment can also vary through time. And so this is work that is led by a PhD student in my lab, Josh Fowler, and is in collaboration with Jen Rogers and Ken Whitney, based at University of New Mexico. And this is where I'll be talking still about grasses, but now focusing on symbioses between grasses and microbes. Okay, so just to introduce the idea of temporal fluctuations, again, I'm going to use Texas here because we all know Texas well. So, if you were here in September of 2011, this is what the drought environment of Texas was like. This was a horrible, horrible drought all across the state. If you take the same time of year in September 2017, very different picture because this, of course, was Hurricane Harvey. So it almost looks like this is a good year, but it actually wasn't. It was just a disaster in a different way in the other direction. And so I've lived in Texas for 10 years or 12 years, and I think I've lived through like the 3 wettest years and the 2 hottest years. There's some kind of crazy stats about all the 1,000-year events that I've lived through in 10 years. So sadly, this is part of our of the planet now, right? So it's expected that these what are called whiplash events, so like abrupt transitions in the environment, are increasing and expected to continue to increase under climate change. So this is really just to motivate the idea of thinking about the ways in which the environment can fluctuate in time. So to go back to our drawing board, we're now thinking about environments that fluctuate. So we can subscript that with T, which makes the vital rates of populations fluctuate, which makes the fitness of the population fluctuate, giving rise to good years and bad years. So we can think about a distribution of good years and bad years, and maybe that distribution has a mean and a variance. And longstanding theory allows us to understand the consequences of that variance. And the upshot of that is basically that variability is bad for fitness in fluctuating environments. And that comes from this approximation, which tells us that the stochastic population growth rate, so the growth rate in a variable environment, is given not by the mean of this distribution, but by the mean minus a penalty that's proportional to the variance in the environment. So variability is bad for fitness, is the sort of general message here. So hold that idea for a second. I'm going to tie in another topic, which seems like an abrupt shift, but I'm going to take it all back together. Okay, so the other topic, of course, is microbial symbiosis. We all know that most macroorganisms, all macroorganisms, harbor diverse microbes that live in or on them. These are some famous examples of coral symbioses and arthropod symbioses. We, of course, have microbes that live in or on us. And ecologists are really just starting to understand the ecological effects of these associations. They're very well described, and we know that there's microbes everywhere, but what are they doing to ecological processes? We work, of course, on plants and specifically grasses, and there's a large fraction of the seed tree or cool season grasses form symbioses with specialized fungal endophytes. These are in the genus Epicloe, and they are vertically transmitted or predominantly vertically transmitted. So that means that as the plant is growing, the fungus will grow through the leaf tissue, and this is what the fungal height will look like inside of leaf tissue. And then as the plant is forming its reproductive structures, the fungal growth will track that and infect the seeds or the ovules that the maternal plant is making. And I don't know if you could see this, but these are fungal hyphae growing inside of seeds. So this is a really, as I said, widespread symbiosis in grasses, and it's broadly thought to be mutualistic. And that's because, or at least partly because of vertical transmission, which of course couples the fitness of the host and the symbiont. So it would be, it's to the advantage of the symbiont to help its host if its host's reproduction is the only way that it can transmit. And that's thought to favor the evolution of mutualism. And there's lots of evidence that these often are mutualistic, but those effects are often really context dependent. And to sort of introduce that idea of context dependence, I'll tell you briefly about this is work that was led by Marion Donald, who is a PhD student in my lab. And Marion did an experiment where she set up experimental populations that varied in their initial prevalence of endophytes in these grass populations, going from only 5% of the plants having endophytes to 95% of plants having the endophytes, and track those through time. And the Y and X axes here are that prevalence in different years. The diagonal line is the one-to-one relationship. So if a point falls on that line, that's a population that stayed exactly the same prevalence as it was planted or the year before. And this was done under two treatments. One was sort of an ambient condition that included a decent amount of drought stress in that particular set of years. And then we had a wet year treatment that actually was designed to mimic a future environment in East Texas where this was done. The East Texas climate projections are actually expected to get wetter. So we wanted to create a wet year that might be what the future could look like. And the main thing I want to highlight here is that you'll notice that the wet year treatments mostly fall along the one-to-one diagonal. And that is essentially a neutral symbiont, right? So there's no change in the prevalence of the symbiont, probably because it wasn't doing very much to its host. And you can compare that to an average year that included drought stress. And all of these points are above that one-to-one line, which means that prevalence is increasing, and that's driven by mutualism. So the more beneficial the interaction is, the greater the prevalence will be. And the asymptotic expectation is that this goes to fixation if there's always increasing prevalence, whereas adding water literally neutralizes the interaction. So that's one example. There's actually many examples of context dependence. So it depends on what environment the interaction is happening in, what kind of fitness outcome you get from the host harboring symbionts. And that is the thread that connects the two ideas that I've talked about, because fluctuations in the environment are fluctuations in context. And so you can get different types of interactions in variable environments due to context dependence. That leads to this hypothesis that symbionts can buffer their hosts against the fitness penalty of environmental fluctuations. And I will kind of talk through the rationale behind this. We go back to a distribution of fitness, of having good years, bad years, and average years. We can expect that the bad years might be the time when it's most valuable to have a partner, if you're a host plant. So we might expect endophyte positive hosts or E+ hosts to have on average greater fitness in bad times than endophyte negative hosts. So the tail of this distribution might not go so low as it would without symbiotic partners. What I showed in the previous slide is that you get essentially neutral outcomes in good conditions, right? So maybe this doesn't change at all. Maybe in good conditions, hypothetically, you could even have a cost to harboring symbionts if you have to pay them. In the grass endophyte system, the plants have to pay the fungi carbon, and they get fungal biochemical compounds in exchange. But if you don't need protection, maybe paying a partner is more costly than it is beneficial. So what I've drawn is a reduction in variance. And because variance is costly, anything that reduces variance could itself be beneficial. So this might be mutualism. Even though the average of these distributions are exactly the same, there might not be any benefit on average to harboring symbionts, but if they reduce the amplitude of your fluctuations, that might itself be a form of mutualism. It might also, of course, be combined with a change in the mean. So maybe it is on average better to harbor endophytes than to not, but you might have shifts in the mean and changes in variance. So we wanted to know, is there evidence for what I'll call variance buffering, which is the reduction in variance through symbiosis? And if so, how does it compare to any changes in the mean? And what are the relative importance of those things? So to answer that question, we have an experiment not in Texas, actually, but in South Central Indiana. And this is work that's funded through the Long-Term Research in Environmental Biology program at NSF. So this is a program that supports decadal scale kinds of questions. And of course, if you're trying to ask questions about variance, you need lots of years to do that. So the experiment was started in 2007 by my colleague Jen Rogers, and it included these 7 host species. These are all grasses that associate with fungal endophytes. And what Jen did was initiate experimental populations by taking founders that are endophyte positive from each of those species, keep treating seeds, which kills endophytes but not plants. And that gives you endophyte negative and positive plants with the same genetic background. So you have an apples to apples comparison to isolate the effect of the symbiosis. So Jen set up about 5 to 10 plots for each of those species. And over the 15 years or so of the experiment so far, we've got over 15,000 individuals that have recruited into these populations, and we track all these, and we visit them every year, record mortality, growth, reproduction, and we've got, something like 30,000 plant year observations. So it's a really rich data set that we can pull a lot out of. And so the basic workflow, as she said, so this is what one of the plots looks like in the field. This is 2.5 meters by 2.5 meters, and all these little tags are run after we worked our way through and tagged all of the new individuals in the population. So we start with data collection, obviously, and then we use hierarchical modeling to variance components in a data set like this. There's years, there's plots, there's species, there's individuals. We want to specifically isolate that interannual variability and the effects of the symbiosis on that interannual variability. And then we can use stochastic matrix models to ask those questions about how important is variance buffering relative to changes in the average demographic performance. So I don't have, there's a lot of data, there's a lot of results from this, and I'm not going to go through all of it, but I want to pick one example just to give you a sense of what data look like and what kind of information you get out of it. So this is one host species. This is Asthuthis subverticillata. It's a native endophyte host in Eastern North America. This is its survival in relation to size, pooling all of the years. So this is kind of the average across years. And first of all, larger plants survive better, and that's not particularly interesting, but we account for size structure. And then the difference here between E plus and E minus tells us that there's an endophyte advantage to survival. So E plus plants survive a little better than endophyte positive. Sorry, endophyte positive plants survive a little better than endophyte negative plants. That's all years combined. And then we can blow that up into each individual year. And I've done that by separating E plus populations up here and E minus populations down here. And I realize there's a lot going on these panels, and I don't expect you to take that all in. What I do want to bring your attention to is if you compare the width of all these lines here to here, there's a narrower width in the E plus populations, and that's variance buffer. That's reduced interannual fluctuations, so that the bad years are less bad and the good years are less good for the E plus populations. So that's one species and one vital rate. And of course, there's a lot more going on in this data set. So I've tried to distill a lot of information into this very simple plot in which species are columns and different vital rate processes are rows. And I've just color coded the sign of the endophyte effect. And so we were just looking at the Asthuthis subverticillata survival, which is a positive effect on the mean and a negative effect on the variance. And all bring your attention to on this slide is that most of this is blue and most of this is red. So on average, we see sort of a lot of evidence for mean benefits of symbiosis, and we also see evidence for variance buffering. We can take all this and integrate it into our stochastic demographic models so that we can look at the effects, across all those vital rates on the mean fitness, as well as the variance in fitness, which I quantified here as the CV. And again, all these species are kind of telling their own interesting story, but for the sake of time, I'll bring your attention to the top part in red, which is the mean across all species, which, as I just mentioned earlier, tells us that there is a positive fitness effect. So a positive effect here on the mean fitness and a weaker but negative effect on the interannual variance of fitness. So that tells us that we have a picture like this of reduced variance, but also elevated mean. So we now want to know what is the relative importance of those things. So I'm going to walk through some cartoonized predictions of what we might see in different scenarios, and then show you the real data that correspond to that. So if we think about the endophyte effect on the stochastic growth rate, which integrates both of those pieces, the mean and the variance, if we have some total effect that's positive, let's assume, because we think these are mutualists. If we do a numerical experiment turning on or off the mean and variance effects, if it's only due to variance buffering that this mutualist benefit happens, then if we simulate only that variance buffering, we should recover that total effect. And if we simulate only the average benefit without variance buffering, we should not get any benefit of mutualism. The reverse would be true if the total fitness benefit is driven entirely by benefits to the mean. We should recover that in simulations that only account for that mechanism, and we should not see anything if we only account for variance buffering. And of course, there's lots of things in the middle where you could have a total effect that reflects a combination of both mean effects and variance buffering. No, there is an interaction that I didn't put on the cartoon because I thought it might be confusing, but it is on the next slide. So there is, and it's actually a very weak interaction, but there is an interaction between mean and variance. So these are the real results. And again, I've got a lot going on here, and there's all species here. I'll draw your attention to the red box, which is the average across all species. Okay. First of all, showing that the effect on the stochastic growth rate on average across species is positive. And we can recover almost the entirety of that positive effect if we only account for the effect of endophytes on the mean. And the effect on variance is very weak and close to zero. So variance buffering happens, but it's not quantitatively very important for the overall fitness benefit in this mutualism. The last thing we wanted to ask, though, is, well, if the future climate is more variable, will variance buffering become a more important pathway of mutualism if for these hosts that are living in a potentially more variable environment? So to answer that question, we did another numerical experiment that I'll sort of walk through here. So if you imagine taking all of the data for variation in fitness for a given host species across years, maybe it looks something like this. We can sample this distribution in a biased way to make average environments less likely. So that's represented here. So what if we took this distribution and sampled only the 6 most extreme years, or the 4 most extreme years, or the 2 most extreme years? When we do that, we can actually inflate the variance, which is shown on the right side here, without changing the mean and without having to extrapolate into conditions that we didn't observe. So we can use our real data. These are real years that happen, but we can just pretend that we're creating environments in which those are the only possible years that can happen. And of course, this is the most variable environment can be that just fluctuates between the best and the worst year. So when we do that, this is the result. And this is set up the same way, but I've now added in those experimental treatments, the numerical simulation treatments. Again, I'll draw your attention to the species mean. And first thing to notice is that when we increase variance, which are increasing green, blue to red, mutualism gets stronger. So there's a stronger benefit of endophyte symbiosis on the stochastic growth rate in a more variable environment, which is cool. And that is due entirely to variance buffering. So you get actually quite a strong contribution of variance buffering in a more variable environment, such that the relative importance flips, where here in the current regime of variability, the average effects are more important than the variance buffering, and in a hypothetical future that's more variable, that reverses. So variance buffering in the current environment is not an important component of this mutualism, but in a future environment, it could or will be. So we find that there's broad evidence across species for variance buffering and demographic buffering. I'm saying that's true in grass endophyte symbiosis, perhaps more broadly. We just rarely have the data to see it, because you need long-term data with these experimental treatments to be able to detect that. A weak contribution of those effects currently, but a more variable environment could be one in which this mechanism of mutualism becomes important for having those hosts cope with environmental stress. Okay, where we're going next with the host symbiotic work is thinking of coming full circle to spatial environmental variation and thinking about range limits, and specifically in Texas. And this is sort of connecting context dependence ideas, which I just talked about through time, back to space. Because if we think that range limits arise because of stress, as you know, in Texas, as you go further west, then that's a change in context in which mutualism might get stronger in more stressful environments in ways that perhaps could extend the ranges of their host events. So we're working currently with 3 host species, and I'm showing these maps here. These are 3 Eastern North American grasses that are endophyte hosts, and they all hit range limits, their westernmost range limits in Texas along the aridity gradient. So we are setting up new common garden experiments, creating populations of endophyte positive and endophyte negative plants across this aridity gradient. Again, coming back to Bastrop, hitting all of our favorite haunts, and asking whether there's evidence for symbiosis having this geographic footprint. So are they changing the distributional patterns of their hosts? Okay. To try to tie it all together and just wrapping up, I want to emphasize I've taken kind of a shortcut in all of this work, which is that I focus on environmental variability, but I've treated the environment implicitly. So I've modeled, longitude and interannual variation, but longitude is not a real environmental driver. It's just a proxy for one. Years are not meaningful. They're only meaningful with respect to how the environment changes across years. So I think the next challenge in our work and as a field is to shift from implicit to explicit treatment of the environment. What is it about longitude? It's probably aridity, but I can quantify that. What is it about different years? And that's important because if we actually want to make predictions about what the future looks like in some no analog environment, we need to actually mechanistically connect environmental drivers to these responses, and we can't just use these proxy variables. So I'm not entirely throwing myself under the bus. I think there's value in what I told you, but I think there is a challenge to then take it further and try to be more explicit about what we mean when we talk about environmental variation. Okay, lastly, I want to quickly just acknowledge the land where this happens. So, I live and work in Texas, and I use Texas as my scientific sandbox, as I told you about. This is land that's the ancestral homeland of a lot of rich tapestry of indigenous groups, most of which have been forcibly removed from Texas. So, I'm still listening and learning about the ways that I could best practice a decolonized ecology. I think that's particularly tricky in places like Texas, where so little of indigenous culture and community has been able to remain. But I think that has to start with being clear-eyed about that history and saying it out loud. And that's really the intention behind the slide. Okay, with that, I will acknowledge particularly Josh, Aldo, Marion, Jen, and Ken, who made important contributions to everything I talked about, as well as lots of other people who made this work possible, lots of field stations across Texas, Oklahoma, and Kansas. And I'm so grateful for their generosity and hospitality, NSF for funding. Thank you for listening, and I'm happy to take any questions if you have them.
First, thanks for your talk. I wanted to ask a couple of questions about the first part. And so you got to this result about basically the female part mattering. So the message then is like that feedback is not there so much, or?
Yeah, exactly.
And I was wondering one detail about it though, because I noticed you use like sensitivity rather than elasticity. That like in proportion to the vital rate rather than just raw changes in the language?
It's a really interesting question, so the decomposition analysis that I showed you only works with sensitivity, and so that's why I used it. I would actually, I think I would expect the same or perhaps a weaker male contribution with elasticity, and that's because the male contribution would be through seed production, which generally, particularly for longer lived perennial plants, has very low elasticities because churning out seeds doesn't matter so much relative to just surviving if you can reproduce for many, many years. So it's a really good question. My guess would be that elasticities tell the same story, or maybe even if it's possible, a weaker male contribution.
Hi, great talk. Do you mind going back to one of your rich slides where you showed the species means? Yeah, that one. I want to make sure I understand this. So for the variance only plots or data points, if it's positive, is that variance buffering or opposite of variance buffering?
Yeah, that's a good question. So, and I'm sorry if I didn't explain it well. If it's positive, it is variance buffering because it's basically a piece of the full effect.
Okay.
So really, these are meaningful with respect to this point.
Okay,
if that
so
makes sense.
you do then have a few examples where you have negative variance, so that would be variance expanding.
Yeah.
Can you like hypothesize under what circumstances it would lead to enhanced variance?
Yeah. You're kind of stumping me. You're exactly right. I go through this and I tell you, oh, just look at this. But no, to be fair, no, there's super interesting stuff happening across species. So I don't know mechanistically, how you get variance expansion. I mean, it has to happen because the endophytes make the bad years worse and the good years better, which is counterintuitive to the way that we think about most mutualisms and symbioses operating. How does that work? Biologically, I'm not sure. The variance buffering we think is through the fungal alkaloids, the biochemical compounds that these plants or the fungi make, which we know can buffer the plants against drought. How is it that the reverse can happen sometimes? I mean, yeah, I'm not sure. I think it's a really interesting question. And where we're going with this is we're trying to actually get into more of a trait-based analysis because we've got really interesting patterns across species. So what is it about one or the other species in which you have a weaker or stronger pattern? Maybe will tell us something more about that. Good question. Yeah.
I don't know if this relates to that at all, but I was curious about the potential for others. So you have this one fungal symbiont that's very interesting, but there are many other potential symbionts, especially below-ground fungal symbionts. So how do you think those? Presumably, a lot of those are horizontal transfers as well. So how do you think that other symbiont effect might be framed?
Yeah, so we haven't looked at it specifically, but others have asked whether there's interactions or interference between symbionts. You can imagine if grasses are paying carbon to support endophytes and they're not giving carbon to below ground microbes, for example. There's actually not very strong evidence for those types of interactions. So I'm not saying they operate fully independently, but we don't have evidence to suggest that there might be some counteracting process happening in other types of partners. Do you have any thoughts of how it might?
Well, I was just wondering if first of all, if the vertical transmission versus horizontal transmission could be part of the story, in that if that would potentially see some where the variance via the vertical integration. But then also whether there would be a context dependence of which symbiont is helping during.
Oh, yeah. And maybe you could answer.
And that's so that's one.
Yeah.
That maybe there were some other. I mean.
Yeah, what we should do is start looking at the soil and like it's still it's going to go for the next 5 years, so there's time. Well, that's a good idea. Yeah.
Thanks for the talk. Actually, like you just mentioned, you probably want to do some like trait analysis. Oh, that's actually what I wanted to like ask. Because well, the thing I want to ask is about because there's so much variations depending on which grass species, and the thinness consequences of the endophytes may differ. So I wonder, I guess one way that the thinness can differ is by the intrinsic. So it's intrinsically each grass may be drought tolerant to different degrees. So I wonder, maybe that's one thing that would determine it. On the other hand, I was just thinking like, how do we, so it seems that, most of the story is told from like a plant perspective, the plant is like benefiting from the, endophytes, but I wonder, like, have you thought about, if the endophytes will somehow like, it seems they're like, vertical transmitted, I guess we're not like switching posts or anything, but I wonder if there might be some selection forces on which grass they like more likely to associate with.
There is an endophyte side of the story, and it's through transmission, and I told you that transmission is vertical, but it's often not perfect. So the plant, and it's not known if this is the plant's decision or the fungus's failure or both, but you often get endophyte positive plants that make endophyte negative seeds. So one of the things we're getting interested in is do we see temporal fluctuations in that transmission rate that might correlate with the demographic effects that we're seeing? So that would be one way in which there's an endophyte fitness component of this sort of fluctuating environment.
Thank you.
Just to follow up on this question, does this endophyte have a free living situation?
No, so they have no environmental reservoir. You can only find them in these grasses.
In different grasses though?
They're exclusive to C3 grasses, but many species of the C3 grasses, and you tend to get different endophyte species and different host species. So there's been like a co-radiation.
Okay, so it's a meaningful question actually to ask what is the consequence of all that for the endophyte?
Oh, sure. Yeah.
Yeah,
Totally.
okay.
Yeah. Because they can't do anything unless their host transmits them.
But they can jump hosts. If they don't want that host, they can jump.
They can't.
The environment is not running.
They can't jump very easily. So it occurs between 1% and 3% of the time that there's any signal of horizontal transmission. They're very faithfully vertically transmitted. Sorry. I don't want to keep. I don't want to keep us. Yeah. Go ahead.
So yeah, just a quick question about it. So the alkaloid is a structural analog of LSD, unless I'm getting a good system. Does it function?
Wait, what's the name?
Is it acting as a defense though? I guess that's about it.
Yeah. So there's so if you've ever heard of like toxicosis in cattle, part of why a lot of people care about these endophytes, which is not the reason I care about them, is because they're agriculturally really important. They can be toxic to cattle because it's LSD basically, and humans use it too. I'm not like we don't do that, but like people get high on ergot alkaloid, and you could find it in these grasses. So they are actually really good at deterring herbivores because they make animals really sick. They also have drought protective properties, but it's less known like how or why. You understand why alkaloid makes an animal sick. Why does it make a plant more robust to drought? It does, but it's not exactly clear why.
These multiple functions though could explain some of the disparate answers in terms of its effect on variance. If it's
Right.
acting as both a defense
Right.
and
Right. Which gets at the point of like, what is the environment? And what's actually varying across years, because that might also be part of that story. Okay, yeah. Thanks.
We just want to thank you. So good to see you.
You too.
Excellent.
Thanks. Great to see you. Okay.
Put it over to Pat. Chapter.
Oh, hi, Norma. I'm sorry to.
Hi,